Guys, I've asked this question before. How many of you guys are Monty Python fans? Yeah. Okay. So, and now for something completely different. Yeah. So, we've just transitioned now from a bunch of information, to looking at scientific papers and how this kind of phylogenetic perspective, trees and microbial populations are addressed by microbiologists to answer interesting questions. So from now on, we're going to be going through papers and talking about how this stuff actually works in practice. And most of this happens according to this graph in one way or another. And so we're going to go through various aspects of this graph in each of the papers for the next few weeks. And so you did this in a way in lab. So you started with a microbial sample, right? In our case, you purify from that particular pure culture. So the microbial sample really is a pure culture at that point. You isolated DNA. Well, in our case, it was trivial. We just picked the colony into the tube, right? You did a PCR reaction. You amplified a ribosomal RNA, and you got sequence data. And from that, you do an analysis. And we'll talk about the term project in lab this week. But this is the part that you're doing in lab. But you don't have to start with a pure culture. You can also do an analysis starting with a complex microbial community. A sample of dirt, a swab from someone's throat, whatever that might be. Isolate DNA, do a PCR reaction, and now this ribosomal DNA, instead of being a band composed of hopefully all one sequence, is a band that may be fuzzy because there's some variation in sizes, and it's a population of sequences instead of a single sequence. And if you've done this properly, then there ought to be some similarity between that population of sequences and the original microbial population, right? If it's half E. coli and half thermus, you'd like to think that half of the sequences here are thermus and half of them are E. coli sequences. It turns out that's not true for a variety of reasons, but you hope that it's approximately true. And then you do something to separate those sequences out, and those molecules out, and you sequence them, so that you end up then with a population of sequences instead of a single sequence. And you can analyze that population and hopefully learn something about the original community, right? And so you might take a swab from skin and look at the relative proportion of all the organisms that live on the surface of your body. We'll talk about a paper where they do that later on. Ultimately, you'd like to know that the sequences you get are actually in that original community. And maybe you want to know a little bit more than just that they're there. Maybe you'd like to know what the organisms look like, or what their three-dimensional organization is within a microbial community. So you can use this sequence information and create what we call phylogenetic probes, which are nothing more than oligonucleotides, usually fluorescently labeled, that you can go back to a microbial sample using fluorescent in situ hybridization, or FISH. These are sometimes called phylogenetic stains, right? They're not stains in the way that crystal violet is a stain, but they're molecules that can be used to tag certain kinds of organisms. And so you might, for example, have sequences here, and you say, okay, this is a really interesting sequence. It's abundant in the environment. I want to know more about this organism. And so you would make a phylogenetic probe against it, go back to the community, and see what it looks like, see where it lives, et cetera, et cetera. Yeah?
Did you say FISH stood for again?
Fluorescent in situ hybridization. We'll talk in great detail about this. So how can you use this approach? Well, one is to identify the predominant microbial group, right? If you take a sample and you get a bunch of sequences, hopefully that composition of sequences will tell you a little bit about the composition of the organisms that you started with. You can actually count them and drop them into phylogenetics groups and hopefully say that the community is primarily epsilon proteobacteria, 10% gamma proteobacteria, 10% beta proteobacteria, et cetera, et cetera. You can also use these in enrichments. Imagine that you're interested in a particular organism. You've done this analysis, you've designed a probe against it. Now you can use that probe in enrichment cultures as a way of measuring your success. It's an assay for how much a particular organism is growing. So you probe all your different variations of the enrichment and see which one is working and go from there. You can use those probes then to go back to the environment and count the organisms. It may be that you get only 1% of them here, but if you go back to the sample, you can actually count them directly and get an objective measure of their frequency in the environment. And you can identify uncultivable organisms. So for example, if you've got a lesion on your skin, there are clearly bacteria there, but they don't grow on anything, then you can use a probe to identify it. You can pull the sequence out, you can do the PCR, you can see what the organism might be, and then, but because it's unlikely to be pure, you get a collection of sequences, you can test them one at a time and see which one is the actual organism. We'll talk about examples of each of these.